First of all, thanks for the invitation to come here and speak, and also I'm quite jet lagged, so I'm going to try to go slow through this. I have plenty of time, I believe, and let's get started. First of all, I want to acknowledge my lab, the Open Behavior Project that we run, our funders, the people involved in the study are listed here, and also an invitation to you. A few years back, Alicia Esquerdo and I wanted to start a Gordon Research Conference on frontal cortex. We were able to finally get it going with support from some people in the room who wrote letters in support of the project. And we ran it last summer in California. It'll run again in New Hampshire in '24. Becca Shansky and Dave Mormon are organizing it. There's a little QR code there if you're interested. Please come, because the main focus of the meeting actually was cross-species issues, structural and functional. And you get reviewed when you have a GRC, and we were criticized to not have enough European participants, so we would really love to have people involved for the next cycle. I'm going to talk today, starting out with a paper that we published in 2019 and give you a 4-year update on this paper. This was written and sent to eNeuro because we wanted to say what we really wanted to say. We wanted to be able to interact with the editors and reviewers and get this paper out in our own way, hopefully with a good sense of humor along the read of the paper. So what, if anything, is rodent prefrontal cortex? The authors were myself, Linda Amirani, who's now a postdoc in Allen Brain, Kyra Swanson, who's now at the San Diego Zoo, Samantha White, who's a 5th-year student looking at a postdoc right now, and also Steve Wise had contributions to this paper as we wrote it. I'm going to take you through some parts of the paper, not the whole thing. The meta-analysis of publications we did. This is not a data-heavy talk. And then we'll talk about rodent brain atlases, and I think that's probably the biggest driver of confusion about what rodent studies really mean for understanding the human brain. I won't talk about a survey of researchers that we did about those terms, and then I'll conclude on that section with a discussion of an idea we had about the corpus callosum and how it may contribute to interpreting functional data in rodents and primates. Then I want to give you an update, and it is the mouse takeover. So we'll show you what's been happening in the last 5 years in publications, the concept of mouse cognition, which is being debated on Twitter more for AI than neuroscience, but there are some exceptions and some caution should be demonstrated. And I want to conclude this session, and hopefully it leads into the rest of the conference, with some comments about what might be a way forward for all of us using common behavioral designs to study these questions. This is the first figure from the paper. Our idea was, let's go on PubMed. We moved to the American University from Yale. We're trying to set up a lab. Let's go and do some homework on what's going on in the literature right now. You know, myself, I've been doing rat prefrontal cortex since the early 2000s. It was not easy to get grants at times because the question was, well, is there really a prefrontal cortex in rat? Why do we care? And as you can see, and I'll use the cursor since the pointer seems to be out, around 1990, there was this explosion of work with the imaging tools that came on, like PET and eventually fMRI, human studies on the prefrontal cortex. Prefrontal generally has meant granular frontal cortex, dorsolateral PFC. That's the word, that's what it usually means. Steady state of work in rodents in frontal areas going back to the 1980s. A lower rate of overall monkey work, but very good work that's been going on the whole time here in orange. And then you'll see around 2000 the takeoff of mouse studies. And so we just wanted to plot it on a log scale. What's the rate of growth? And mice are basically going to pass rats by 2020 in terms of total papers. That's happened. I'll show you that later. Actually now passing humans within this decade. And the other thing we did was, since we downloaded all the papers, we looked at their abstracts and keywords from the National Library of Medicine, where there's librarians that actually verify these are the appropriate keywords for a study. And we just started to do word frequency analysis. And I had to teach a class on Python for our undergrads and grad students, and word clouds are a fun way to learn how to code. We still do this in our class every year, and we've shared the code, and people are using these for their websites. So you can find it on our GitHub. But what they show you is the frequency in which these terms pop up in the literature. And so we categorized it by species, general species. We categorized things into different colors for different types of terms. But what jumps out right away is that while rats and mice, the research has been on some pretty common topics, things like dopamine, nucleus accumbens, hippocampus, behavioral studies, genes. You don't see those terms in human or monkey word clouds. They're actually quite divergent. And even really the human and monkey topics don't really converge. The second thing in the monkeys is that the words are much larger. There's a lot more variability going on in the monkey research compared to human, rat, and mouse research. So we're not on the same page, and one of the main points in the paper is this meeting, we need to come together to talk about what we can do to work on common themes. And if our goal is to understand the human prefrontal cortex, what can we do together to advance the field to do just that? Now, this is probably why the paper's been cited a whole lot. And if you focus on panel B, what we wanted to do was deal with this PFC nomenclature in rodents. Here's two examples of the Paxinos Atlas from 1998 and from 2014, when it was remapped into the Brodmann's areas working with Brent Vogt, and numbers were used to replace labels that had been used before. The labels are in the mouse atlas from Paxinos, they're in the Allen atlas. The eBrains is great, by the way, for teaching people 3D anatomy. The same terms are there. But what's happened is, as we said, well, basically the precise locations and pretty close sections change. It's very hard to understand how these then relate. We've seen already DMPFC and VMPFC. It's one of the worst nomenclatures to use because prelimbic cortex gets split up or included in one and not in the other. It's very confusing. It's very confusing to primate researchers trying to read rat papers and mouse papers when you use those terms. So this is one of the points in the paper that we made, and I want to just step through the points here one by one. The first is dual citizenship. Many labs have said MPFC. Right? And first of all, we've acknowledged there's no lateral PFC in rodents, so why do we even use the letter M? But MPFC is one term, and then ACC is the other. And if you go and look at the ePhys literature, you'll see people with electrodes and tetrodes in the same places saying it's anterior cingulate cortex, and others saying it's MPFC. And generally, they mean prelimbic cortex, or the ACD, or CG. There's a whole other nomenclature, and it's really a mess. So part of this dual citizenship, these two terms for the same area, we plead to please show where your probes are in papers, show us a diagram of the actual anatomy, provide summary diagrams where you actually put down all your recording sites or your viral tracers and show the distributions, and give us your stereotaxic coordinates in your papers. And it's actually changed since this paper's come out. There are now even tracer studies, which many people don't report their coordinates in. We're seeing it in more recently. So it's having an impact, we think, we hope. The second point is alphabet soup and lines in the sand. Prelimbic, ACD, CG1, CG3. It's a mess, and if you don't work in the rodent, how do you know what it means? And the lines in the sand, just in these two atlases, the lines change. Now, Area 33 pops up. So it's very confusing to people, and it's not made it easy for the field to move ahead. All right, then I went back to my college exam obfuscation, right? So let's make something really clouded, beclouding, and abstrusity. There are papers that have been published and tweeted about that use phrases like the prelimbic prefrontal cortex, the infralimbic prefrontal cortex. What is that? It doesn't exist. The prelimbic medial prefrontal cortex. So please don't do that. Just say prelimbic cortex. It's much simpler, and people know what you mean. We don't really know why people do it, but it continues to show up even in the past year on Twitter. And finally, the main point was, why can't we be friends? One thing that you could just say is medial frontal cortex. And Nod, who was here, it was in his talk in Developing Humans. Just go with MFC. In fact, that idea came to me in a talk I gave informally at Yale in 2011 on this topic, where Dave McCormick said, just drop the pre, the problem goes away. I think it's true. It's a simpler way to do things, and it gets rid of the confusion. If you're precise on what you report and you don't try to oversell it, we can probably understand what you did. The point of the paper that we thought would have a little more impact, and maybe hasn't as much because it's usually been cited to talk about the areas and how to map them out, was our proposal for what they may mean functionally. And this slide is busy, but I'll take you through a few parts. The first general observation was that brain shapes differ between rodents and humans. And so in the lab, we'd use our fists. Rat brains are like a kind of clamped fist where you're doing a knuckle punch, whereas in the human, you're making a full-on fist. And there's a curvature that differs between the two species. The brain is generally flat in the rodent with a little L on the front of the callosum. You can measure its angles. You can spend time looking at its geometry. Kyra Swanson, who was a mathy person in my lab, spent a lot of time looking at this. And then you can get examples of human collosa and make measurements of the curvature. And you'll see quite a dramatic difference in the angles of the callosum. Now, the callosum's a lot of white matter. It's going to exert a lot of force on the developing brain. It may constrain how neurons migrate in development, leading to different positions of cortical areas. And without getting into panel C, if you look at the three places where most rodent labs work, prelimbic, anterior cingulate, and below in purple infralimbic, they sit in an L around the rat or mouse callosum. And if you just take those 3 points and rotate them by the measured angles from humans, they shift around the callosum into areas that are pretty much the mainstream macaque areas of medial frontal cortex. The anterior mid-cingulate cortex, also known as DACC, kind of like CG, area 24. The pregenial ACC, very similar to prelimbic now in its position, and infralimbic cortex becoming subgenial ACC. So just looking at the angle of the callosum, you can kind of start to see where these areas might be. And then if you think about what you want to do to understand them in rats and mice, maybe we should look at human studies, for example, the Rushworth group, who's looked at things like foraging on these areas, and try to come up with simple studies we can do in rodents to relate to those studies in humans, and then try to work together, bring in fMRI and bring in some EEG, and really try to get this stuff moving ahead. That was our main proposal from the paper. I want to extend it, because this paper's been invited as a TINS article for this year, to what we're thinking about going forward, and that's this point. This is our pandemic review that we wrote about a whole series of studies that led me to go from sustained bar presses to studying licking and reward, and now we're back to studying decision making. But if you look at the differences in the anatomy of the rostral and caudal parts of rodent frontal cortex, you'll see really two distinct kind of networks. The rostral MFC is what we propose is a feed and breed system. It's very autonomic. For example, prelimbic cortex is massively connected with the insular cortex in a bidirectional way. As you saw earlier, its projections are to the ventral striatum, nucleus accumbens. Parts of the thalamus, the paraventricular thalamus, a hotspot in recent years in publications, very, very different projections coming from rostral and caudal MFC to this region. For many years, we've known from the Price Lab in WashU about massive differences in the connectivity in hypothalamus, feeding-related centers particularly, orexin, et cetera, being picked up from the rostral MFC, and periaqueductal gray. But nothing really going rostrally to superior colliculus. Also nothing really visual going into that rostral MFC. Whereas in the caudal case, you've got a system that really dominates to the superior colliculus and things like dorsal striatum and more activating fight and flight, fight or flight systems within the hypothalamus and periaqueductal gray. So a real trade-off between these two areas, one of which could be, as we say at the bottom of the slide, time spent in foraging behaviors versus time spent in vigilance behaviors. There are very few species who can simultaneously effectively search for food and be aware of a threat. There are some birds who are able to do this on a beach. They can turn their heads sideways, open up their vision to look for something that might strike them from the air, another bird, while they pick up food off the beach. But most of us aren't capable of doing that. If you're really going through some shrubs and you're a rodent or a human gathering food, you're probably checking out the quality of the food, and you're maybe not aware of a threat near you. You have to stop foraging, look around, see what's happening. So that trade-off exists, and perhaps this circuitry mediates it. The second point of this story, which we're going to develop as we write the next paper, is that the areas right next to these regions are motor regions. The rostral medial frontal cortex right next to it and further forward ends up being the head in rodents, neck muscles, facial muscles, jaw opening and closing. The areas that are back here comprise bilateral control of the forelimbs, the shoulders, unilateral forelimb control, and hind limb control. So there's a body map in the motor cortex sitting next to a map of something on the medial frontal cortex, and maybe it's the related systems, modulators and pathways to the brainstem, controlling things like arousal and feeding. That's the basis for understanding the organization of this circuit. If you think about it this way, you can design experiments with long arrays of electrodes and start to record across this axis, and that's something that we're starting to do in our lab, having tasks with threat and reward and varying the value of those things to try to figure out how this axis works. Lots of focus on dorsal ventral, not a lot on rostral caudal. So that's the proposal. Let's all start thinking about that dimension to understand how rodents relate to humans. And it goes back to the previous slide with the rotational scheme, CGPL moving around the rotated callosum. That's the main point from the original talk. So here's data from March 1st, downloaded off PubMed. Orange is mouse, blue is rat, red is human. Since 2015, since this paper came out in 2019, but since 2015, there are more mouse papers than rat papers per year,
And in fact, in the last couple of years, more mouse papers than human papers that focus on prefrontal cortex. So I think it's really important to take a pause and think about what people are doing in these studies when they do behavior. And on the right, the cumulative plots, you can get rates of growth. By the end of this decade, there'll be more mouse papers than human papers. Here's just really quick the mouse papers from 2014 and from 2022, and the only real difference is stress and social. These words are now very large. Of course, what this means for stress is hanging a mouse by its tail, dropping it in a bucket of water. Is that the stress we go through every day? Not to poke too much fun, but social interactions, putting two mice together in a box, is that really normal? So, I mean, we need to think about what people are doing when they have these translational effects and relate to cognition. And again, just last year's data, human, mouse, really not a lot of connectivity between them. You don't see words that are common. We're still not doing a good job bringing the communities together on these topics. We need more meetings like this. And then there's mouse cognition. So I'm gonna do it. There's some stuff off Twitter that I think we need to talk about, particularly this tweet on the left. It's a nice study. They can relate it to ApoE4, look at how it might affect things like transport in axons. Restoring transport restores mouse cognition. But what was the mouse cognition? It's a novel object recognition procedure. It's got 11 controls and 14 I believe animals with the treatment, the effect size, or the p-value P0466. There's a bit of overlap here. It's a nice study. I looked at it. It looks a nice study, but hope and AD is hashtags. My mother had dementia. She died from COVID. Telling people that you're going to cure Alzheimer's disease based on something like this is misleading people. You're giving them false hope. It's not something we should be doing as scientists. So let's not do this on Twitter. Let's report the results and talk about it. And it's great that there's progress, but let's not say we're going to cure Alzheimer's based on a mouse study. And down below, a lab has come up with a great platform for phenotyping in animals, and it uses T-mazes and two object recognition tasks. These are basic cognitive procedures. So things do exist, and there is hope. In a paper written in 2015, hope for studying cognition in mouse. More ways of assessing it, and what I love is the Western University's project with touchscreens and mouse bites. Now you can really do set shifting in cognitive studies. So there are tools to do this, but I think we need to be careful about it. And object recognition clearly is impaired in Alzheimer's and dementia, but it's not the whole story. So folks need to be a little more precise and careful. Doing the same kind of analysis for mouse cognition, it explodes even more. There's a massive number of papers from mice talking about cognitive processing. Again, just spiking around 2000, and here's the word cloud. Alzheimer's models are dominating. It's not prefrontal, it's hippocampal. So we need another meeting to kind of bring the people together on this one. And we'll see where it goes. But there's, in my opinion, concern here over the massive focus on mouse models for studying Alzheimer's. Okay, so just to end, I'm over by a few minutes. Here's what we originally wanted to write the paper, what if anything about, and for a number of reasons this part kind of got dropped out. The idea being we need simple behavioral designs to work together to make progress. So typical designs in rodent studies, linear tracks, T-mazes, visual discrimination learning, commonly called decision making in these days, head-fixed mice on trackballs. It's all for valid reasons. Lots we're learning. The technology is incredible in the last 20 years. But human studies still use complex visual stimuli that mouse studies really can't work with. Often the designs include language, words that the participants react to. That's not going to work in mice. Long delays in the trials. Most mice can't mediate long delays, nor can rats. They're not made to. They're prey. Multi-stage trials. Very, very hard to train in a rodent. Takes a long time to build up to. And dependence on working memory and verbal instruction. And actually verbal instruction would be something that would be wonderful to do human studies where you don't instruct people and just look at how they learn. And there are people doing this. But that's what we do in animals. So why don't we try to get there? So simpler designs are needed. Reaction time tasks, my lab, that's what we used to use. We're getting back to it. We collaborated with Michael Frank, Jim Kavanaugh on a human study. They did our rat timing task. We got the same EEG and LFP, and the people involved in that study, Nanda Kumar Narayanan and Jim Kavanaugh, have continued to collaborate related to Parkinson's disease. They're working on dopamine therapies based on that now. So simple designs can be used. I personally think we need to start looking at how things change with the task acquisitions that we use, because the basis for most rodent studies are early learning. Outside of people that do electrophysiology, the classic lesion work and other kinds of studies that are done, it's instrumental learning. Boline and colleagues, this is stuff we need to think about in our designs. Task events should be precisely timed, so comparisons can be made between in vivo measurements and behaving rodents and brain signals in things like EEG and MEG. It's very difficult to relate fMRI to spike activity, but if we have parallel designs in humans that use imaging, EEG, that's my seat, hi, that's my seat, thank you, and perturbation methods, maybe we can start to bring this stuff together. So that, of course, then means that funders need to take this into account and make funding available for people to really work together and try to understand the basis for these different measurements that we do. So this is the general idea, to try to come together, keep it simple on the behaviors and start to have more collaboration and to work with our funders to give us the resources to do that. So that's what I wanted to say. Thank you.